Hi, my name is Jessica Redline, and today I'm going to be demonstrating Lab 5 for micro. It is 8-1-20 for LAPU, micro 225. Let me see if I can position this a little bit better. Okay, so with this one, we're going to be doing differential stains. And so I have all my slides labeled over here. I already cleaned my surface. I have my E. coli, my S. epidermis, and my S. cerevisiae. So they're all labeled here with a dime size underneath it. And I'm going to, I have my inoculation loop that I already disinfected in my alcohol cup that I have here. And I placed it in there for about 30 seconds, and I'm gonna remove my loop and place it on a clean paper towel. So first I'm gonna start with E. coli, and I'm gonna show just one step of this on camera because it's probably gonna take a while for me to do the experiment, but I'm gonna show the full steps of this one. So this is the E. coli culture that I've been having in the fridge. So I'm gonna remove the top to this one, and I'm gonna pass it through my flame that I have down here. Let's see, I have my fire down here. Pass it through just to sterilize it. Okay, I'm gonna insert the loop into the broth carefully. I don't want to touch the sides. Kind of move it around. And I'm gonna get that swab and I'm gonna rub it against my slide. That's E. coli. Like any excess that's on there. And then I'm gonna place this tube over the flame again. Sterilize it before I cap it. Okay, I'm gonna place this on the side. So I already transferred the liquid that was adhering to the loop onto the E. coli slide, and I replaced the cap. I'm going to place the loop into diluted bleach, sorry, prior to disposing. And this slide is gonna completely dry. So I'm gonna let it dry a little bit, and I'm gonna do the same for my other cultures now. So next we're gonna do S. epidermidis. I'm going to sterilize the ring of the tube with the flame. I'm going to get a clean inoculation tube. Pass it through the tube. I'm going to put any excess onto the dime size spot. And place that in bleach. I'm going to pass it through again before I cap it. And place this on the side. I'm gonna so my E. coli and my S. epidermidis side have both been had their cultures put on. My last one is my S. cerevisiae, which is right here. I'm gonna open it, pass the tube through, get a new inoculation loop. going to pass the loop through. And I'm gonna swab my S-Service-A slide with any excess that was left onto the loop. Put that in bleach. Going to redo this one and cap it. So I'm pretty sure my E. coli and my S. epidermidis have cooled completely. I'm going to place one sample in my staining tray. We're going to start with E. coli. So you can see it there. My E. coli in my staining tray. And I'm going to add 1 to 2 drops of my crystal violet. Oh, I'm sorry. You know what? I actually forgot. I missed one step. Before we place it in the staining tray, we're actually going to clamp the slide and we're going to pass it through the flame. I'm not holding it directly over the flame, but just passing it through about 5 times. So I'm going to do that now. I'm going to let that cool. I'm going to get my S. epidermidis and do the same thing. I'm just passing it through. I'm not holding it directly over. Okay. Again, my S-Service-A has dried completely as well. So I'm going to pass this one through the flame. Okay. So let those cool completely. I'm going to blow out my flame because I don't believe I need it for the rest of the experiment. So I'm going to blow it out for now. And place that to the side. Okay, so I'm going to start with my S-Service-A. Again, I have it in my staining tray here, laying flat, and I'm going to add, sorry, my glasses are fogging up. I'm going to add 2 drops of the crystal violet. Well, it says 1 to 2 drops. So, just like so, right into the dime center. Okay. I'm going to let that sit for approximately 60 seconds. And then I'm going to gently rinse the slide with tap water over the staining tray. And then any of the excess contents, we're going to let it slide down the middle of the drain, and then we're going to return the slide back to the tray. So, and then after that, we're going to be doing more drops of the PVP iodine, the decolorizer, the safranin, and so forth. So we'll do that with each slide, but I'm only going to show all the steps for this first one just because it's going to take me a long time, and I don't think the video is going to upload with a really long video. So this has been about 60 seconds, so I'm going to go ahead and walk over to my sink and start rinsing this. Okay, now I have my paper towel and I'm just gonna let it sit on top of that and like lightly let it sit there so it can dry. And so it says to wait a little bit after I rinse the slide. Okay, now I'm going to add 2 to 3 drops of the PVP iodine. And you put it back in my tray. Okay. And it says to wait 60 seconds before we gently rinse the slide and return it to the staining tray. So we'll do that, and then we're going to add 2 to 3 drops of the decolorizer and the safranin. And then we're going to place the slide in the paper towels to completely dry. So let me walk over to the sink so that I can get started on rinsing this. It's almost been about 60 seconds, so I'll be right back. Okay, now that we're back from that one, the PVP iodine, we're going to be doing the decolorizer. And that is this clear one right here. This liquid one. It says to add 2 to 3 drops. Okay, and then we'll wait about, this one says 30 seconds. So we'll go ahead and do that. I'm curious to see what the outcome of this is going to be, considering that we're staining it with all these different stains. I'm not sure how it's going to look under the microscope, but I'm interested in seeing what it would look like. So I'm going to walk this one over to the sink and gently rinse it, and then we're going to be on our last stain of the safranin. Okay, now that I'm back with that one, I'm going to do the last one, which is the Safranin Graham stain number 4. And I'm going to add 2 to 3 drops. Okay, this one's like a reddish color. So this one, it says to allow to wait 30 seconds. And then we can gently rinse this one. And then that concludes the whole Graham staining process. So after this is complete, I'm going to rinse this off and let it air dry completely. And I'm going to place it on the paper towel until it's done doing so. And then I'm going to repeat all the steps for the remaining slides. So for S. epidermidis and the S. cerevisiae, I'm going to repeat the same steps and processes. And I'm going to allow them to all dry completely. I'm going to upload my photo and take the picture with my name on it, and we are going to see these slides under the microscope at all powers. So we'll start off at low, and then we'll go to medium, and then the high, and then the oil immersion to see which one we are able to visualize the cultures the best. So I will upload the next video with a follow-up of the results.